So we'll move to our next presentation. Dr. Tim Widmer. He is the USDA National Program Leader for Plant Health. He is the point contact for all USDA and ARS projects related to plant health and is involved also with the microbial resistance, soil health, invasive species, and issues related to agricultural biosecurity. He's been with USDA-ARS since 2000, working as a research plant pathologist in France and in Maryland. He received his PhD in plant pathology at the University of Florida, Gainesville, and had a postdoc position at Cornell University. Dr. Widmer's presentation is titled Lessons Learned from COVID-19 Can Be Applied to Plant Disease Epidemics. Sorry. Dr. Widmer?
All right, hold your breath, Gary, and I'm going to try and share my screen here and get going. So just let me know if everything looks okay. Okay?
Perfect. Thank you.
Great. Well, thank you very much for that introduction, and I'm very happy to be able to give this presentation today and be a part of this meeting, bringing so many experts on peanuts together. Yes, I agree, Gary, that although an in-person meeting is better, I too think you made the right call in holding a virtual meeting. As you said, holding virtual meetings, you can have so many more participants, international participants, but it does take a lot of work to put these together. So I appreciate those behind the scenes that are doing this work, but I think that the number and the quality of the presentations and the posters is impressive across so many disciplines, which is a tribute to this organization. This is my first APRES meeting, but I have to say I'm feeling a little bit I'm missing something of those peanut munchies, and so that is one thing I may look forward to in the next live meeting if I can attend. Well, one of the messages that I've been emphasizing this past year and a half in particular is that plants get sick too. Now, I don't want to minimize in any way the human impact and the effects that it's had on us, but we have to point out that our agricultural commodities are just as much in danger. Over the years, we have done such a good job in maintaining the food supply that these dangers are often not on the minds of the average consumer. However, with the pandemic, I think food shortages brought about this more to the forefront, and we now just need to help the public make that connection between plant diseases or plant pests and food security. And the reality is that we lose a lot of food and agricultural productivity through plant diseases. The FAO estimates up to 40% in global production is lost to plant pests, with an economic impact of $220 billion annually. For peanut, there are over 70 known pathogens, more than 15 are known viruses, and more are being discovered. Some specific examples that I found for peanut include yield reductions associated with foliar diseases such as leaf spot form the greatest threat of all diseases to the southern United States. Early in leaf spot can potentially cause annual losses of around $600 million. In Florida alone, on a statewide basis, annual yield losses attributed to peanut leaf spot vary from 5 to 40%. And yield losses to white mold is heaviest where peanuts are grown each year or every other year. Estimated losses in these fields may reach 20% or more of expected yields. Tomato spotted wilt virus is one of the most damaging viruses of peanut. This virus causes a serious disease of peanuts in Florida and was first discovered in the state in 1986. When it first arrived, peanut losses were in the tens of millions of dollars annually, as susceptible varieties were commonly planted. And I found information that yield decreases of greater than 50% are still observed in some times. And also nematodes are widespread and destructive pests on peanuts. For example, each year in Alabama, 5 to 10% of potential peanut yield is estimated to be lost due to nematodes. Well, this list here is all familiar terms that the public has heard over this past year and a half during the pandemic. Gary led into my presentation perfectly, as he already mentioned some of these terms in his earlier presentation. However, these are also terms that we can associate or use with plant disease epidemics. So what I'd like to do now is just to go through each of these terms and give some examples of research as related to plant diseases. And since I've been working with ARS for over 20 years, I'm a little more familiar with those research programs. So most of my examples will be based upon that. However, this is not to say that there's not just as important and relative research going on in universities and private sector, as we will hear about in this meeting this week. In fact, many of the examples that I'll be giving are in collaboration with these partners. Okay, so I'm slightly stretching the terminology here a little bit on masks to include row covers. And I realize that they are impractical for peanut production, but the concept is the same, and that is to prevent or limit the plant spread. It's a physical barrier. For many viruses, the key is limiting or controlling the insect vector from infecting new plants from an initial source. It also may mean planting density so that the microclimate is adjusted to more non-favorable conditions for plant infection or rain splash or crop rotations to reduce inoculum load. This would also include controlling weeds. I'm sure that everyone is well familiar of the role that weeds play in the disease cycles, as they can be reservoirs for viruses and other pathogens. For example, in this publication from Kill et al. In 2020, tomato spotted wilt is known to be harbored in many winter weeds. And one of the difficulties in identifying which weed hosts are reservoirs is that many of them are asymptomatic. And so it must laboriously be tested for which ones may be an important alternate host. But since many plants infected with these viruses do not die, the inoculum can continually spread if not managed properly. Quarantines are an important part of keeping plant pathogens out of the US or further spread within the US. USDA-ARS has specific regulatory policies for certain pathogens. And although at times it may seem restrictive to us researchers, these regulations are put in place to protect US agriculture from invasive pathogens. And I'm also including here as voluntary quarantines, where we purposely avoid working on the pathogen here in the US for fear that it might escape. There are still ways that we can do research on these pathogens, and one very effective way is to set up international collaborations. And one important component of this international collaboration is to set up field trials to screen for resistant plant material where the pathogen exists under natural conditions. Not only do we avoid the risk of release, but we get more accurate results under these natural field conditions and inoculum sources. An example that I'm going to use here is peanut smut. This pathogen is native to Argentina, not yet found in the US, but it is a serious threat with yield reductions reported up to 70%. ARS has set up field trials in Argentina to screen US germplasm and have found some resistance. And you can find more information on this by reading Kelly Chamberlain's poster during this conference. And for ARS, we also, at ARS, we also have a very unique biosecurity level 3 facility at Fort Detrick, Maryland. And this is an APHIS approved facility that can safely work on any pathogen or isolate, regardless of its origin. Recently, Congress awarded ARS new money to build a new facility with the lab and greenhouse capacity, which should be breaking ground fairly soon. I encourage you to seek collaborations with them if you are working to expand your research to include exotic pathogens. There are always opportunities that we're looking for. Diagnostic testing during the pandemic, that the public became more familiar with the importance of accurate diagnostic testing. This is something in the plant world that we have recognized for some time. Accurate, reliable, easy to use, and quick diagnostics are critical for determining the pathogen that is impacting the crop. Without it, you may not be applying the proper management strategy, or it may delay management enough that the pathogen can spread further. Some of the tests that are available, like for COVID-19, are dipsticks and PCR. And a more recent PCR technique, as seen here on the right, is called digital droplet PCR, which is basically analyzes micro drops of DNA individually. It's more sensitive, and it's reported in some cases to be 100 times more sensitive in detecting a viral genetic material than the standard PCR technique. Dipsticks are probably what most would strive for, as they are simple to use and do not require any special equipment. You can get a result in a matter of minutes. However, sometimes they are for more general identification and may not give the specificity that is desired or needed. But as a quick diagnostic, they are a great first approach that can at least narrow down for what is causing the issue. Well, along with testing, maybe you have seen this article or heard something similar, that the dogs are able to sniff out COVID-19 and could be used as a diagnostic or detection method. Well, at ARS, we've been using canines also to detect plant pathogens. We've been successful in using canines to eradicate plumpox virus from the US and also help detect citrus canker and citrus greening. Canines are perfect as an early in-field detection system. Accurate sampling can be an issue depending upon the disease. I'm using citrus greening here. It's a perfect example where the pathogen is often not distributed evenly throughout the canopy. And so collecting enough samples to avoid this error can be almost next to impossible. Collecting and analyzing physical samples in this case can easily return false negatives if the infected tissue is not selected. But for citrus greening, we found that the canines can detect the pathogen in the tree much quicker than typical molecular methods, such as PCR, and much more than any visual symptoms of the disease appear on the tree by detecting volatiles released by the pathogen itself, as shown here by the cartoon in this graphic in the center. To put this sensitivity in perspective, dogs are capable of detecting 1 drop of blood in the volume of about 3 Olympic-sized swimming pools. It's I like to say it's an old technology being used in a new way. Well, there are other applications for canine detection as well, including tomato chlorotic spot virus, which is an emerging tospovirus, like tomato spotted wilt virus in Florida. However, again, don't let the name fool you. Like tomato spotted wilt virus, this pathogen also infects peanut, as Bob Kemmerite from the University of Georgia found in Haiti. It has not, to my knowledge, been found on peanut yet here in the US. I put those last 2 slides up really to build up back to using the canines to detect viruses in vegetables. This can also have application for peanuts. We have an area-wide based project from our unit in Fort Pierce, Florida, which is training canines to detect chlorotic spot virus in vegetables and weed hosts. And here's a video of a dog in training to recognize pepper plants infected with tomato chlorotic spot virus. In this training, the plants are in pots in a grid with known inoculated plants randomly distributed to measure accurate detection, false positives, and false negatives. The dog checks each plant and sits down when it detects the pathogen infecting it. And it is then rewarded by a few seconds of playing with the ball. As I mentioned, research is showing that the dogs are detecting the pathogen itself and not volatiles released from the infected host plant. A dog can rapidly go through the entire field, and it may also just be necessary to go around the periphery of the field, depending upon the epidemiology of the pathogen. I would like to add, in a similar concept, that some recent technology is also incorporated using handheld devices to detect signature volatile compounds. And I think this technology has also a lot of potential. So we also have heard a lot about COVID-19 variants, especially the Delta variant that now seems to be so prevalent and spreading further. But plant viruses also have variants that can change the way they spread and infect. I show this slide because resistance breaking tomato spotted wilt virus isolates are now known. And you can see the different reactions here in the left in tomato to those different variants. And these variants highlight also the importance of diagnostic testing. Variants happen because of rapid replication, natural selection, and resistant plants with resistant genes are the most effective way to stay ahead of this arms race. And here you can see the effect of incorporating a resistant gene in against tomato spotted wilt virus. The SW5 gene on these plants here on the right, which is the resistant gene against the virus, and then the plant there on the left that doesn't have the resistance gene and it is infected with tomato spotted wilt. But an arms race occurs between the plant and the virus as resistance breaks down can happen by only one nucleotide substitution in the virus as it adapts as well. And in 1983, a severe strain of peanut mottle virus was detected in Georgia, and it caused severe devastation on what became susceptible peanut cultivars. Resistance has always been the strategy that is touted as the most effective and durable against losses caused by plant pathogens. However, this can be time consuming and take years before plant material is released. And this is one reason why it is critical to assess and predict which plant pathogens might be of importance in the future. And I'll get on that a little bit later. Traditional breeding techniques are still the most commonly used, but now are being incorporated with molecular genomics to determine which genes are important. And at some point, genetic engineering may also need to be considered. And who knows what the future may hold as this field is really just starting to expand and the technologies are really quite amazing. Just thinking back in 5 or 10 years of how much advanced we have done in this work. The next couple of slides are from Corley Holbrook and his work in Tifton, Georgia with USDA-ARS. I'm not going to go through into much details as he has a presentation later on this week, but here you have released several cultivars with near immunity to the peanut root knot nematode. The first release was Tifguard, which eliminated the need for nematicides even in fields with heavy nematode pressure. They then used marker selection, assisted selection to combine nematode resistance with the high oleic trait and released the cultivar on the left of the slide. Last year, they released 2 additional peanut cultivars with nematode resistance. They're currently also using molecular marker assisted selection in an accelerated backcross breeding program to integrate major genes for resistance to leaf spot. Growers currently spray 6 to 8 times per year to control leaf spot. We anticipate that these breeding efforts will greatly reduce and perhaps even eliminate the need for leaf spot sprays. And again, attend Corley's presentation on the 15th to learn more details. I wanted to show this slide from Baoju Gao, also at Tifton with ARS, as it highlights just the time and effort that it takes for breeding to get enough seed just for testing and verifying that the seed contains the resistant genes that we're trying to incorporate. And you can learn more about Baozu's work in his presentation later this afternoon. So understanding the mechanism is also very important. Here in this example for tomato spotted wilt virus on tomato, you can see here on this graphics what is happening in the resistant line. In a more susceptible line, the virus can cross in between the cell walls. But however, in the resistant line, the cell-to-cell movement is blocked due to callus deposition. And vaccines are also very familiar terminology now, but a lot of people are not aware that the concept of vaccines protecting plants is similar to what occurs in humans and animals. We don't necessarily inject the vaccine into the plant that is shown here in this illustration, and plants do not produce antibodies toward the pathogen per se, but an immune response by the plant is put in motion. And we normally call this cross protection. Citrus tristeza virus is a great example of a success in the past in using cross inoculation or immunization. It was found that a mild strain of the virus, which was taken from a healthy tree shown here by the arrow, could be co-inoculated with a severe strain of the virus and protect the tree. and get normal production from citrus, even though it was infected with the virus. And it's important to study the mechanisms involved in this cross protection so that we can exploit it for plant protection and for other pathosystems. I was also made aware that there is a commercial product available for cross protection against Pepino Mosaic virus using the mild strain of the virus. I'm not familiar with this product and do not know its efficacy, but I just wanted to point out that there are possibilities that growers can easily use. And who knows what other potential commercial applications could come out of this work. Interference RNA or RNAi is also becoming a research topic that is just being explored for its possibilities to protect plants. You're probably already well familiar with it, but just in case, I wanted to show this simplified graphic of what normally occurs, where through transcription, DNA will produce a messenger RNA strand that then translates into a specific protein. However, when a specific RNAi is applied, it interferes with this messenger RNA by binding to it and thereby blocking the translation of the protein. An example that I'm going to give here is an application to kill a feeding insect that could be a vector for a pathogen. This preliminary experiment was successful when RNAi was applied as a soil drench. It was taken up by the plant, and feeding insects ingested the RNAi that interfered with some aspect of their life cycle and thereby killing them. And you can see that there on the right of the photo of the dead psyllids. And I'm sure that you're all aware of the issue of labor shortage. Even before the pandemic, labor shortage was an issue, especially in the agriculture sector. And the US Bureau of Labor Statistics estimated that employment of agricultural workers is expected to increase just 1% from 2019 to 2029. But one way that ARS is approaching the labor shortage is to investigate how robotics or automation can help alleviate the labor shortage and also advance precision agriculture. For peanuts, Rebecca Bennett at our Stillwater, Oklahoma unit with ARS is collaborating with Oklahoma State University and looking at ways to evaluate peanuts, in this case for peanut smut infection, using X-ray technology. Screening peanuts visually can be very labor intensive and not always accurate. However, using this or similar technology along with artificial intelligence can more accurately test and identify diseased peanut pods. The next slides are not examples from peanuts, but they have similar applications of what can be used or already being used for peanut production. We're also adding automation in pest control measures that are more precise. And here we do field mapping for disease and pathogen pressure. So that the farmer only applies the chemical where it is needed the most, thus saving money and the environment. Field mapping can be done by taking representative samples throughout the field or by imagery collected by satellites or unmanned aerial vehicles or drones. These images are then translated into prescription maps where hotspots locations are identified and communicated directly to the farming equipment through GIS that determines an appropriate amount of pesticide to an exact location depending upon those hotspots. In this example for cotton root rot, it was demonstrated that growers can save over $10,000 in a single year on a 93-hectare field in pesticide costs alone. We're also starting to incorporate drones into different farming system applications. Collecting data from satellites can be costly and dependent upon the flight path. But drones are more flexible and can be used to fly over and take images of large fields in a fairly inexpensive and timely manner. These images can be processed and presented to the grower for what they are needing. In this example, drones are being used to detect herbicide-resistant weeds such as Palmer amaranth. among row crops. For an output, the field map will highlight the Palmer amaranth weed, and using hyperspectral analysis can determine whether it is glyphosate resistant or not. In this case, the farmer is able to successfully identify glyphosate resistant Palmer amaranth in soybeans with this drone-based remote sensing system, and be able to respond appropriately by using different mitigation strategies. System is able to correctly identify a Palmer amaranth weed as glyphosate resistant or not about 90% of the time. Pest management also has opportunities for incorporating robotics. The USDA is collaborating with companies such as Trick Robotics to develop autonomous units for pest control options. Here is a robotic prototype in a time-lapsed video going through a commercial strawberry field for demonstration purposes. This machine will be fitted with a light, ultraviolet light, which is found to be effective in pathogen suppression. In order to be fully effective, the UV light needs to be applied at night, as shown in the second video here onto the right. These types of applications are a perfect example of how robotics can be incorporated into modern farming for autonomous practices, and also protect workers by reducing exposure to hazardous conditions, such as being exposed to UV light or pesticide applications. Okay, so after showing the similarities in the language, what now have we learned really that can be applied to agriculture? Well, the first thing is really a surprise to those of us working in agriculture, and certainly not to our farmers, and that is we are vulnerable in the agricultural sector. We already know about food deserts, but during the pandemic, we saw disruptions in the food supply chain and harvesting. And this was reflected all the way to the store shelves, as shown here from photos that I took at my local grocery store about a month after that first initial shutdown. These shortages have also been highlighted in the news media, and peanut butter was one of those items. Now, I love peanut butter, and I know how hard it was get. I have to admit, I prefer crunchy. And if I was the APRES president, I'd be pushing crunchy over creamy. But I don't want to take away, from those who like creamy. And now my handlers are saying I'm getting off script here, so I need to get back to my presentation. But peanut butter is an important part, as you can see here by this headline. It's considered a staple, and we saw shortages in peanut butter. I think it really highlights the importance that this industry brings to the consumer. But this comes back to one of my first slides that plants get sick too. Often I think that we are really just one plant disease epidemic away from other catastrophes. And I've been trying to emphasize that point as much as I can. I don't want to be an alarmist, but it's taking the opportunity to get others to make that connection and to plan ahead for what we've learned. And we need to be proactive rather than reactive in our research. And this can be risky because we do not know for sure what will be the next plant disease epidemic. And we have limited funds and time, so we need to assess it properly. In ARS, we have a program called the National Plant Disease Recovery System. And one of the purposes of this program is to put research funds towards new and emerging pathogens, many of which are not yet here in the US. But this gives us the flexibility on a yearly basis to start some initial research on these pathogens. It is really, it is just a Band-Aid per se, as per the funds available are just enough to get a project going. However, important research has been conducted through this program. Variety screening for resistance against peanut smut and diagnostics to detect Indian peanut clump virus are a couple examples that have benefited from this program for peanuts. However, funding is limited, and there are many new and emerging diseases that deserve attention. So how do we predict the next epidemic? Well, we need to set up a global surveillance and monitoring system for one. And this needs to involve international collaborations and a multifaceted collaborative effort within the US, involving government, both at the federal and state levels, universities, private industry, and also growers. We need also to continually advance our research for more accurate and reliable diagnostics that can give quick results that we can react appropriately and quickly. And finally, we need to start screening for resistant plant material now, especially for some of the pathogens that are not in the US but are threatening. And you can see that all of these are intertwined with each other and will take a collaborative effort. So I just want to acknowledge my ARS colleagues for providing me with a lot of the information that I presented here. Having this opportunity, I'm going to highlight and encourage you to check out their presentations and posters later in this conference. And if you ever get to Beltsville or Washington, D.C. Area, please feel free to come by and talk with me. We're always wanting and hoping that our stakeholder input, it's very important to ARS. I've also listed here my contact information, so please feel free to contact me if you have any questions or ideas. I'll be here for the rest of the morning session, and if there's time to answer questions now, I'm happy to take, I really thank you for your presentation.
Thank you, Dr. Widmer. Great presentation. Of course, when you started talking about the crunchy peanut butter, I'm that fan as well. So, and the next time when we're having our APRES meeting in Dallas, we certainly would like to invite you to come and join us and enjoy a lot of the munchies that we have. I think that a lot of your presentation really reflects back to what APRES has as a goal, in that it's not only that research in frame of what we do, but it's also that educational part of what we do, to take what we can research, to take what we can learn, and not only bring it to people in our industry, but to take it to everybody outside of our industry and let them know the things that we're doing. So excellent presentation. Questions for Dr. Widmer? I'm not seeing any that are on here. I know that one of the things that you were talking about was the use of the canines and the use of the handhelds. Is there any other type of new technology that we might be just on the forefront of?
There are, especially this handheld volatiles. I think some of these are now starting to even add with the smartphone, so they're easily accessible to everyone with a little attachment. And when you talk about sequencers or PCR machines, and those too I've seen attached to cell phones, and the technology and the size of the technology, I think are just we don't know what the future will hold. I think in the next several years. So those are some of the things I think that we're really excited about.
Excellent, and I absolutely agree. I think that whole technology of having that phone in your hand and things that you can plug into it nowadays, it's not only taking your O2 reading, it's taking your blood sugar readings. There's just endless ways that we can be able to use that. Some questions have popped up here. One is really a comment here from Chris Butts saying, we will be glad for you to bring the other national program leaders to Dallas with you. Wonderful. So we certainly encourage you.
Yeah, I think most of you know Roy Scott very well. He's been involved with the peanut group, I think, for a long time. So.
Perfect. Soraya asked or said, wonderful presentation. What is the stage of the peanut quarantine? When will we be able to import peanut for research?
Yeah, great question. And, I'm not the person to answer that, but my understanding is that we're trying to work on ways to safely be able to do that. I think the issue is right now, we don't want to import peanuts into the area where peanut production is happening because of a potential accidental release could be devastating. And so we're trying to avoid that. And we're working on solutions. We've had some talks even to maybe, how we can do with Fort Detrick with the BSL-3 facility. But they are limited as well, but we are trying to work on solutions like that. Thank you.
Perfect. Thank you very much. Another question, comment from Dr. Colbreth. Your inclusion of the situation with peanut smut was an indication, but how important is it for us in the U.S. to monitor plant peanut problems worldwide?
Well, I think it's critical because the earlier we know about these problems, the earlier we can start doing research on them before they come to the US. And it comes back to my being proactive rather than reactive. So like in peanut smut, we already know and have identified some resistant varieties. So if, and I think unfortunately when it arrives in the US, we'll be able to at least have some foundation for supporting the industry and moving and hopefully avoiding any real catastrophes. And then it goes with a lot of the viruses that we don't know yet that can infect peanut. We really need to be proactive in our research. I think it's critical that we study these peanut diseases. But again, I recognize that some people may say, well, how do you know, or which do you work on because of limited resources and personnel? And you just hopefully have to do proper assessments and your homework to make your best guess and work from that.
Absolutely. I think that that partnership that we have not only within the U.S. but globally to be able to pinpoint those possible diseases and possible cures. We're a stronger force as a team than we are as a single individual. A question here from Charles Chin. Is there any organization like WHO to work on plant diseases worldwide?
Wow. Yeah, great question. There are the CGIAR centers, and there's the Peanut Center in India, I believe, ICRSET. But there's not an overall arching, and that does make it a little difficult, for coordination, collaborations. But hopefully, leadership that can then seek out these collaborations, and that's probably right now what we have to do. But there are FAO, for example. CABI out of United Kingdom has the disease database. So there are resources that we can use to help helpfully collaborate and coordinate these research.
We have another question from MD Ali. Nice presentation. What would the national strategy or policy to work with foreign what would the national strategy or policy to work with foreign collaborators? Is there any funding support available?
So with ARS, we do highly encourage foreign collaborations, just as the examples I've shown, because we find it critical. One of the other unique things about ARS is we actually have 4 international laboratories that are associated with ARS. One of them is actually owned by ARS. It's in France. It's where I started my ARS career. Now they normally work with biological control of invasive species, pathogens or weeds and insects, but we have worked with pathogens as well. We also have an ARS associated lab in Argentina, one in China, and one in Australia. Now, we can and we easily do set up collaborations internationally. There's a process, through material transfer agreements and the like. But this is usually just a formality, and we can easily set up collaboration. Funding, well, there is base funding within our projects, and they have the freedom to use that to set up for collaborations. The program that I mentioned, National Plant Disease Recovery System, it often is used to set up collaborations in other countries for this type of testing. So there are funding opportunities. They're not as much as I would like, of course, because I think it could be expanded, and there's a lot more potential, and we should be utilizing those collaborative efforts more.
True. There was a chat here from Kelly. It said APS is currently working on a global plant pathogen surveillance system, which would go back to that question that Charles Chin had.
Yeah, that's right. They're trying to set that up. I think a lot of it is being based out of North Carolina State University.
Perfect. Any other questions? I see that we have 122 people that are attending, and we all appreciate your time and this presentation. It certainly brings that coordination and that likeness between what's going on in the world with the human aspect in coordination with what's going on globally in the agricultural and within viruses and diseases within agriculture. So excellent presentation, and really brought all those points together. Thank you very much.
Thank you. My pleasure.